Hi everyone, in this video I'll be going over Lab 4, Microcontact Printing and Contact Angle Measurements. As mentioned in the previous video, there's a lot of pre-lab reading this week, so it takes some time to go through that material. On a different note, I do want to bring to your attention that your first set of lab reports are due on the 22nd. The templates and report questions have already been posted in week 3. I would recommend going through these prior to the lab section so that you can ask your TA for help. If you need additional help outside of section, you can email me or the other TAs to schedule office hours or attend Dr. Silence's scheduled office hours on Friday. So with those announcements out of the way, let's get to the actual lab. Remember that this is the last of your 3 lab series, and all the way back in Lab 2, you made these PDMS stamps. In Lab 3, you fluorescently labeled BSA in order to make a fluorescent pattern. Now, in Lab 4, you're going to combine these 2 components in order to complete the lab. And what you should end up with is a window-shaped sort of pattern that's green under exposure to blue light. One thing to be aware of is that you won't be able to do this with your materials as is. You'll need to actually treat your PDMS to make the surface conducive to stamping. And based on what you've learned about plasma in Lecture 1 and protein adsorption in Lecture 2, you should be able to figure out why plasma oxidation of your PDMS aids with protein patterning. As a hint, think about it in terms of relative surface hydrophilicity between the different surfaces. 1, between your stamp itself, and 2, between your glass. Think about this and discuss with your TA in section why you think plasma oxidation aids in protein patterning. So to start off, you should have 2 stamps per group. For those sections who have only made 1 per group, the TAs have made extras for you to use. In accordance with this, your TA is actually going to split you guys up into 2 different groups. This is because we can't have everyone doing 1 at a time, so it just makes sense to split you guys up. For both the parts of the procedure, however, you will need to plasma treat your stamps. For microcontact printing, you're going to need 1 whole stamp. While for the contact angle measurements, you're only going to need half surface treated. The treatment, however, only lasts around half an hour, so you'll need to move your samples very quickly after treatment. For microcontact printing, it's imperative that you handle your stamp carefully, especially after adsorbing your protein onto it. When you're using your Kim wipe to remove the liquid, make sure you do never touch the surface and only get the liquid off. In addition, I want to emphasize how important it is to make sure that your stamp does not completely dry out. If it does, you'll be unable to transfer your protein efficiently. One other thing to point out is that because you're using a fluorescent microscope to visualize your pattern, you're going to want to minimize photo bleaching. So use foil when necessary and dim the lights in the rooms when you're handling your solution. For those of you who are starting with the contact angle measurements, remember that you only need half of your stamp plasma treated. This part can be a little tricky when you're trying to take pictures, and I personally found it helpful to hold a black object behind the droplet for best contrast. In addition, when I'm actually physically placing the droplets onto my stamp, I'm going to try to avoid the edges because these can cause unintended edge effects. So kind of want to keep it in the center, and you don't want it here. Let's make a big X so you remember that. Make sure you take at least 3 to 5 images per surface. That way you can get statistical analysis to determine the contact angle for your surface with water. That's all for this video. As you write your lab report, think about how you might use the techniques you've learned in these labs for your independent projects. I'll see you guys next time.